Now we switch to the biochemistry of sialic acid, and I would like to, let's say, present you two data set. One is very old and one is very new and unpublished. And I will start with our location. So who doesn't know where we are? Halle is here, Berlin is here in Germany, and this is around 180 kilometers southwest from Berlin. It's a very old university. The name Martin Luther University indicates already that Martin Luther was professor at our university more than 400 years ago. Okay, so what is sialic acid? Most of you, or probably all of you, will know that sialic acid is a sugar. And this sugar is needed in our body, not for energy production. Normally, sugar is, of course, or the name sugar is related with glucose, and glucose is used for ATP production, so for energy. Sialic acid, not. Sialic acid is more or less a structural component of glycoprotein, also glycolipids, and the extracellular matrix. Sialic acid is not one sugar, it's a family of sugars. What you see here, there are more than 50 members of the sialic acid family in nature. Fortunately, they are not all present in our body. In humans, the major sialic acid is N-acetyl neurominic acid. You see here is the formula. And in most mammals and other animals, we find the major sialic acid, which is N-glycolyl neurominic acid. I will come to this in a minute. Now, on this, or with this cartoon, you see the sialic acid. So sialic acid is a part of glycoproteins. You see here, here's a glycoprotein, and here this purple star. I can go back. Let's see here, here's the purple star. This is the normal sign of sialic acid. This purple star is the outer sugar of all glycoproteins, more or less of all. They are built in a certain way, and many glycoproteins have here sialic acid and also glycolipids. So in nature, this looks like this. When you have a cell here, here's a membrane. So this is I can't see it. So this is the intracellular part. Here's a membrane, and all this fluffy stuff here are these glycans, and the outer glycan, of course you can't see it, but the outer glycan is sialic acid. Okay, now we come to the biosynthesis, and this is not my scheme. So this is a scheme of German Chu. Published this in a paper, but you see here a cell, and of course, sialic acid is not a vitamin or something like this, so we can synthesize sialic acid by ourselves. And of course, since it's a sugar, we start with glucose, so the cell must or uses glucose, is transported into the cell, and then many steps will follow. And the first sugar, which is really important for the direction into biosynthesis, is N-acetylglucosamine, which is glucnag. But this is activated by nucleotide, UTP. So the activated sugar is UDP glucnag. From UDP glucnag, we get an epimerization. This means only one hydroxyl group is in another position. And we get mannag, N-acetyl, manosamine, and the UDP is cleaved off. So the energy for the epimerization comes from this cleaving UDP. And then in the next step, we have a phosphorylation of mannag to mannag 6-phosphate. And then only one step follows. We need phosphoenol pyruvate, PEP, you see here. And then we have the phosphorylated neurominic acid, which is then dephosphorylated, and that's neurominic acid. Interestingly, and this is very important for the activation, because when we have these glycans, you see here, these sugars, the sialic acid, must of course transferred on glycoproteins, and this happens in the Golgi. But before this has to be, or sialic acid has to be activated again with a nucleotide, CTP, which is a very, very rare activation. So most sugars are activated with UDP or GTP, but not with CTP. And this happens, at least we believe it, in the nucleus. So we have the activated, and then we have the sialyl transferases. We have many sialyl transferases to build these structures, and then we have these glycoproteins. One important point, to which I will come later, we have a feedback inhibition of the activated sialic acid, so of CMP sialic acid, to the GNE. So this here is, of course, the GNE. It catalyzes two reactions, the epimerization and the phosphorylation of MANAC. And this is a feedback inhibition. Okay, now I would like to introduce one other point so that we have different sialic acid in mammals. So humans, as I told you, so this is the major story of Ajit Waki, you see here. So the major sialic acid in humans is N-acetyl neurominic acid, or NU5AC. But all the other animals have NU5GC. So this is a hydroxylation you see here. And sorry, not here. You see the hydroxylation, of course, it's a position here. See, it doesn't work. Okay. But anyway, it's just a hydroxylation. And the interesting fact is that we, meaning humans, lost the enzyme activity 2 million years ago, more or less. So now it's working. We have not an active enzyme. So this is a major difference between mice, or at least other mammals, and humans. Okay, now to the data. Very old ones. So this is the original start of my not really scientific work, but at least my postdoc time. We created a GNE knockout mouse. And this was before we knew that there's GNE myopathy. So our interest was just to find how important is sialic acid for life. And this knockout, this was a classical knockout done by Martina. This is more than 20 years old, the picture, Martina and myself. So this was a really interesting project for us because we never did molecular biology before. But we were successful and could generate a classical knockout mouse, and this was lethal. So proof of principle, sialic acid is necessary for life. So you see here the original data from this old paper from 2001. We had an antibody which we produced ourselves, of course, unfortunately. But you see here GNE in the wild type cells, and in the heterozygous cells you see only half, and in the knockout cells you don't see anything. And cells means, since the animals couldn't survive, these were embryonic stem cells, which we derived from embryonic day 3.5, because the animals died at embryonic day 8.5. So we could generate cells at least. And you see on the right side the quantification, relative quantification of sialic acid of these cells in the presence and absence of serum. Serum contains a lot of glycoprotein and also sialic acid, of course. And you see the difference that the wild type cells have a lot of sialic acid and the knockout cells not. And that there is a difference between culturing in sialic acid rich medium and nearly no sialic acid medium. Okay, now this is, I think, the last from the old data. We tried a lot of sugars, monosaccharides, to restore sialylation. And since at that time we didn't know that GNE has two enzymatic activities, we used, of course, MANAC, because MANAC is the first product from the epimerase reaction. And you see here a Western blot with an antibody which recognizes polysialic acid in these cells, in these embryonic stem cells from the mouse. And you see what you expect. So the wild type express it, the heterozygous less, and the knockout cells, there's no polysialic acid. And in the presence of MANAC, and this was the day or the hour where the MANAC treatment idea was born, more or less, we could restore polysialylation, and not with glucnac. And glucnac is very similar. It has just another conformation of the hydroxyl groups. This could not restore polysialylation, but MANAC could do it. Nowadays, and also some years later already, we asked how the MANAC is phosphorylated, because the GNE is missing. So the MANAC kinase is of course also not there or not present in the cells, but this works. And in most cells we analyzed up to now, and even in whole animal, the MANAC treatment works. So you see that MANAC can be metabolized to sialic acid, even in the absence of GNE, and we do not know why. And now I would like to present you some unpublished data. And I'm happy that I can show this data to you, because the mice which were generated by Stella, Trani Rosenbaum, are really new. And these are now knockout mouse or GNE knockout mouse in specific organs. So she created a knockout mouse in muscle and in liver, and in muscle and liver. And this knockout was induced by tamoxifen some weeks, not some, 6 weeks after birth, because she wanted to prevent the embryonic lethality. And what you see now is now very interesting. You see here the knockout and the control, so the wild type always, so knockout in blue and control in whatever, orange. And you see here this complete block and this somehow striped block. And you see that this is the N-glycolyl acid. This is the major sialic acid of mice. And you see a reduction in the muscle GNE knockout, a reduction of the major mouse sialic acid. But these mice had a lot of N-acetylnucleic acid, which is a human sialic acid, more or less, and there's no difference between. So this is very interesting, and we cannot really explain why the N-acetylnucleic acid remains more or less the same. You see that there's no effect. So after knockout in the muscle, there's no effect in liver and also not in kidney, for both for N-acetyl and N-glycolyl. For the muscle and liver knockout, the effect is more dramatic. You see here the quadriceps. So the knockout has very, very low N-glycolyl acid, but still the amount of N-acetyl is the same. And the same is in liver. In liver, you see some differences in the N-acetyl sialic acid, but of course you have a dramatic reduction in the liver knockout and a low or not reduction in the kidney. And again, in the kidney, you have a lot of the human form of sialic acid, which in principle shouldn't occur. And we have no real explanation for this, why this N-acetylnucleic acid is present in such high amounts. And what is a bit, let's say, disappointing also is that the mice have no phenotype at all. So no muscle phenotypes. They are completely healthy after 12 months. So they have no weakness. And even the immune system and everything seems, I mean, they are not analyzed in detail, but they look pretty normal. And this is, for me, really very, very surprising, because I promised Stella before that the liver knockout is lethal, but it isn't, as you see. Okay, so what I wanted to tell you is that the complete GNE knockout is lethal, at least in mice. That humans have a unique sialic acid. And I'm just in time, very good. Yeah. And that's a knockout in muscle and or liver in mouse has no phenotype. And we are still in progress, or we have to do a lot of work to find the reason for this. And with this, I'm ready for questions.